warm welcome to all the distinguished scientists who are here. I think it's a very special day for us. We have been used to completely filled auditoriums 2 years ago, but COVID has actually changed and defined a new normal. So in this context, I know that today Professor Amitabh, when I requested him to give the first lecture as a part of the distinguished lecture series, it was something that was running in the back of my mind. Can I approach him or not? I mean, whether it crosses the line of audacity, but he's been very kind. He has been a mentor to us. He knows the institute probably more than before I did. So we're very fortunate to have him. So, in fact, a very warm welcome to Professor Amitabh Chattopadhyay, Professor Redana, Professor Prakash Babu, and also apologies for informing you very late. There was some kind of an arrangement issue in the live streaming we were trying to figure out. And all my colleagues from DRILS, scientists, students, and other researchers. I believe we also have many people who are also distinguished scientists from other institutions who are also joining students and faculty from CCMB and other IITs as well as other CSR institutes and University of Hyderabad also. So I think a warm welcome to all of them. And the context for today's just let me give a small preamble about why we started this AIC. Professor Redana, he has been actually championing this academia industry at different levels, including at BioAsia and at the FABA level. But what we have seen is in the last couple of years that having a strong hospital orientation, again, something that you've also been advocating, having a clinical context makes the entire discovery much more channelized, more oriented towards patient-centric or basically addressing unmet medical needs. And the world has become so complex, therapeutics and the disease scenario has become so complex that one doesn't have a late-stage way of coming with solutions. One needs a very fundamental discovery and need to fast-track, accelerate this innovation to take it, again, to a patient who desperately needs such an intervention. So in this context, last 3 years, in fact, I'm very happy to share that we have actually built our hospital collaborations. We have a good collaboration with AIG hospitals. We have been working with other cancer groups within the country and outside. And also with Manipal, with Kasturba Medical College, we have started the program in rare diseases. So the clinical contributions and impact has been defining most of these. So slowly but steadily, I think as DRILS, we have basically migrated into a scenario. Of course, the blessings of government funding is needed, but closely working with academia, industry, and clinic on some challenging problems and working with a lot of other institutions and collaborators. So I think that's where it is. Last 3 years, I can just, to substantiate my example, I can say, first and foremost, Professor Redana is here. So we have been working with Professor Redana on the psoriasis program. I personally, when I see this entire program, I think the clinical impact is enormous, and it is basically striking at an area where the other psoriatic treatments have not really looked at it from a mode of intervention. Then there have been other very important repurposing programs. Professor Prakash Babu himself has been a major part of it, where one of my colleagues, he works on the area of diabetes, looking into other additional indications, more from a repurposing context. And we also have a very important initiative called the DRILS Rare Disease Initiative, where my colleagues have been working, Dr. Aarti, Kishore, and Kiranam, using zebrafish models. So when we started 3 years ago till today, we are happy to share we have created models which should enable a drug discovery mode with either repurposing and others. And all these things, there are several clinicians who have been very gracious to work with this institution, who have visited us, who have contributed their thoughts. I strongly think that now the time has come to add a C to the AI. Again, Professor Redana is the one who can only champion it at a national level. But in that context, we thought we will basically have scientists whose work has really been very inspiring and also sets a context for all the younger generation to take inspiration and follow suit. So in this, let me read out actually what we have placed as a part of our flyer. So this DRILS AIC Distinguished Lecture Series celebrates the path-breaking contributions by eminent individuals whose pioneering work has a strong impact on addressing unmet medical needs, scientific solutions that impact society at large. The paradigm now is very clear. Whatever science we do has to impact the society. And what better can teach us the right lesson than the COVID? Last 2 years, COVID has actually taught us how to operate under fairly challenging scenarios, risking our own individual self even by being in a laboratory at the same time with low oxygen levels thanks to the mask. This is all a challenge, but last 2 years have seen whether it is a great effort by CSR and all the IITs, University of Hyderabad and other universities, to some extent DRILS also is, our ability to accelerate innovation contribution has probably seen a much different pace, even at the policy level, decision making and others. So what I see is the accelerated innovation that we see in the 2 years could have some learning for us to maybe make it a norm to pursue it on a daily basis. So it was in that context we thought there is a lot to look back into the COVID scenario and see how we have been able to contribute and how do we take those learnings. With that, again, Professor Amitabh, I think thank you so much for this. This is again, you're coming back to home after a long time, but we look forward. And also specifically Professor Redana and Professor Prakash Babu. There is a slide, this is something that I was also hoping for, so I'll just maybe request Professor Redana, who has been like a mentor to me also, just to share some couple of thoughts, because this meeting, organizing this meeting has been more of a psychological challenge than more of a physical challenge for all of us. And we have industry representations from Origin today, so I will request Professor Redana and also Professor Prakash Babu, who have been collaborators, mentors, and also well-wishers to this institute. Again, I think many of you knew the institute even before I joined, so once again, I will just request a couple of minutes of your thoughts and also your guidance as to how do we really look at this, and then we all eagerly look forward to Professor Amitabh's lecture. I also encourage the students to be able to ask more questions and take general guidance, because there is a reason why we are pursuing this line of science, right? There is a patient or there is a societal benefit which far exceeds our individual roles and positions. So I think I hope that you'll make the best use of it. And yes. So Professor Redana, I'll request you to please share your thoughts and
Let me first thank Dr. Srinivas, who surprised me. So I thought I came here to just listen to Dr. Amitabh's lecture, but he also asked me to say a few words. So this institute was created with the hope that it forms the bridge between the academy and industry and come up with innovative discoveries like the well-established institutes like Scripps and other institutes. And slowly I think that really helped many of the scientists in the university and other academic institutions also to come together and interact the thing. We have been talking about academy and industry interactions for past more than 2 or 3 decades, but not much has taken place. Even today also we had to talk, and it has reached a stage of where a clinic is required in order to revitalize this think process also. And in fact, on the same lines, only just last week I had a discussion with Lanka wellness leaders. Srinivas Lankaji served in the industry, and he has been one of those thought process leaders in the country also, in mainly in the pharma sectors. So when I proposed that why not we have a kind of forum where we could promote academy industry interactions. So specifically, many of the industries, they do not know what the infrastructure that is available in the academic institutions, what kind of expertise is there, what kind of leads that are available, and at the same time the academic institutions, they do not know what is the need in the industry. and the requirements of the industry, and there is need. So, though we have been promoting these kind of interactions through the Federation of Asian Biotech Associations, which we started in 2005, and BioAsia is one of the flagship events of that. And very recently we started FABA Academy to bridge the gap between academic industry in skill development activities. And as part of that, we have been conducting a series of lectures and workshops. In fact, tomorrow there is a workshop on biosafety and biosecurity and its biomedical applications in diagnostics or hospital setups. And so, like that we have been doing, but what we thought was to bring the industry and the academic institution to a platform and then showcase what is available. In addition, a number of startups have come with the support from the government of India through BIRAC and other things. They also have leads, and what they do not know what is the next phase of these kind of leads that are there. Bring them also into the forum and take them forward. So, we have decided to have maybe in the next month sometime a kind of interactive meet, and then propose a series of such kind of meetings which will probably promote the academy and industry. So, I do not want to take much of the time. I wait for Amitabh's lecture. Thank you for the opportunity.
Very good morning, all of you. I thank Dr. Srinivas, Dr. Parimal and Dr. Kishore for inviting me for this talk. In fact, I came here for two purposes. One is it's a long time to see this institute. After COVID, this is first time that I am visiting, got an opportunity. And also I did not see Professor Amitabh for a long time. So, I wanted to come and listen to his lecture. He is not only mentor to you, he is mentor for everybody, in fact, especially for the neuroscience people. We always, without remembering him, we do not conduct any conference. So, he is such a nice man, he is down to earth actually. So, I am happy to be here to listen to him. So, normally in our university, when we go to the class and all, hope our university students are not here, right? Okay, right. We conduct surprising tests, surprise test. Today we will come and say that you have to write exam. So, maybe because we have some other engagements and all, we wanted to conduct exams. So, Dr. Srinivas did the same thing by inviting me to talk something before I came here to listen to him. So, the reason, one of the reasons could be, so I am associated with this institute for a long, maybe from the inception onwards. As you know, for everybody's career, everybody's life, when they are going for climbing the stairs and all, will have the turbulent period. So, the turbulent period does not mean that the ship has to sink, but it has to come out in a victory. So, I can attribute that one to the ILS because of the leadership being provided, and the people that they are associated with this ILS has emerged as one of the important component of the system, not only to the University of Hyderabad. So, we are proud to say that ILS is part of us. DRILS now it is part of University of Hyderabad. So, because of their enormous contribution to the research and collaboration and encouraging and joining with some of our students, and also allowing students and faculty members to join them. So, that is very nice to listen to this and visit once again. The other one I would like to point out is, as Professor Reddana already indicated, that industrial collaboration. I will talk about the other side of the coin. So, it is a translational research. Now, any research, especially in the biology, maybe even in the chemistry also, biology, once unless you do we call it as the word must have come long back, it is a bench to bedside. Unless the research that you are doing in a bench translated into a bedside for the patient utilization, so then the basic research should be translated, otherwise it will not have a real meaning. So, I am happy that Dr. Srinivas and his group, I know all this group I am also contributing little for them. Recently we had one paper in cellular oncology. That paper is a small paper, but the concept was treating the patient using the pantoprazole. So, pantoprazole, as you know, that it is proton acceptor and all. So, the concept was the tumor microenvironment is always be a it's acidic environment. So, that is one of the reasons for the drug resistance. So, we have taken that clue and we worked with that and we showed that it is a wonderful. So, if we also used the temozolomide which is the anti-cancerous agent. So, this enhanced its properties. So, we presented before neurosurgeons and neurologists of KIMS. So, then they appreciated that it is really surprising for us. So, that is the reason, see, unless we repurpose the drugs, unless we go into the details, the doctors may not be realizing it. So, even in the doctors also nowadays, they are also realizing that. So, basic research is equally important. That is the reason why hospitals are now even coming to us and to have a collaboration. Another point I would like to mention here is, right now I am the Dean School of Medical Sciences. As you know, unless we perform, it will be perished, any school or any department or any education. So, COVID also taught us certain things and we started a course on infection prevention and control. We started a diploma with a little worry that students may take up or not, but now the participants are doctors, microbiologists, they are all joining and they wanted to join. Even 50 is the upper limit we kept, but they wanted to join for next course also. So, that is the another thing. The second one is we also started, that is with Infection Prevention and Control of India, Dr. Rangareddy, we are doing it. It may emerge as the regular course also in long run. Other course we started with Apollo, it is a critical care. So, critical care for the, as you know that many medical colleges have come up, even nursing colleges have come up where there are no facilities at all. So, when the patient admitted in the crisis, they do not know how to intubate the patient. So, all these trainings is kind of upskilling the program. So, we started that. So, what I would like to emphasize is the, unless we adapt to the nature, unless we adapt to the circumstance, so it will not good for the system or the science. So, therefore, I really congratulate and appreciate Dr. Srinivas for taking the initiative and also my colleagues here present, I think Kishore is here. We are all doing certain things which are actually useful for this. So, useful for the translational research, and I wish that this should really grow in a longer way and accommodate much more and many more diseases and take hold of the hospitals and all. Therefore, it will be really nice to take part into the second line of the research in the translational research. So, I congratulate once again and I thank Dr. Srinivas and two of my colleagues here for inviting me and to take part in this. So, thank you very much.
An immense honor to have Professor Amitabh Chattopadhyay present with us today, and it is my pleasure to introduce him to all of you. Though it is a difficult task to list all of his achievements, I would like to mention a few here. Professor Amitabh Chattopadhyay is an eminent scientist working in the areas of membrane and receptor biology and biophysics. He is presently a SERB Distinguished Fellow at the Center for Cellular and Molecular Biology, that is CCMB, in Hyderabad. Professor Chattopadhyay's work is focused on monitoring the organization, dynamics and function of biological membranes in healthy and diseased conditions, and he is known for developing the parallax method for determining membrane penetration depths of a wide variety of membrane-bound fluorophores, including proteins and peptides. It is these pioneering studies that have led to a better understanding of the dynamics of hydration of membranes and proteins. Professor Chattopadhyay received his bachelor's degree in chemistry from St. Xavier's College, Kolkata, and a master's in the same subject from IIT Kanpur. He obtained his PhD from the State University of New York at Stony Brook and was a postdoctoral fellow at the University of California, Davis. Subsequently, he joined the Center for Cellular and Molecular Biology in Hyderabad. Professor Chattopadhyay is a fellow of 3 National Science Academies of India, that is FASC, FNASC and FNA. In addition, he is also a fellow of the World Academy of Sciences. He also holds the position of Fellow of the Royal Society of Chemistry and Royal Society of Biology. In 2001, Professor Chattopadhyay was awarded the Shanti Swarup Bhatnagar Prize in the Biological Sciences category by the former Prime Minister Atal Bihari Vajpayee for his outstanding research achievements. He is also the recipient of the prestigious J.C. Bose Fellow in 2008. Professor Chattopadhyay won the World Academy of Science Prize in Biology in 2016 for his seminal contribution in understanding the role of membrane cholesterol in the organization and function in healthy and diseased conditions. He is also the recipient of Professor G. N. Ramachandran Award for Excellence in Biological Sciences and Technology, presented by the Honorable President of India, Shri Ram Nath Kovind. Professor Chattopadhyay has served as the founding Dean of Biological Sciences at the Academy of Scientific and Innovative Research. He is also a distinguished visiting professor at IIT Bombay and an adjunct professor at the Special Center for Molecular Medicine, JNU, IISc, IIT Kanpur, TIFR, Mumbai, IIT, Indian Institute of Science, Education and Research, Mohali, and the Royal Melbourne Institute of Technology, Australia. He is also an honorary faculty member at Jawaharlal Nehru Center for Advanced Scientific Research in Bangalore. He is an author of over 300 papers and has nearly 14,432 citations to his credit, with an H index of 64 and an I-10 index of 219. We are privileged and proud to have Dr. Amitabh Chattopadhyay begin this lecture series for us. Thank you.
Thank you, Srinivas, for this invitation. I am privileged to be the first non-virtual or physical speaker in this series, and I hope this continues, because all of us would like to, Dussehra just went, so we want to get past the dark days and go to the good days. Although the dark days, as Prakash and Professor Reddanna said, has taught us a lot how to make opportunity out of a challenge. Before I start my talk, just 1 minute I'll take, because I have been associated with DRILS for a long time, even when its name was not DRILS, just like Professor Reddanna. And Professor Reddanna talked about Scripps Institute, but this institute was initiated with a great vision from both leaders and scientists, and a lot of people came together. One of a kind in India. In 90s, most of you young people don't even know the ecosystem in 90s, and the idea that you could do academy and industry together those days was almost unheard of. And one of the model was Whitehead Institute, and University of Hyderabad came because you need to have a strong academic background. You cannot just do collaboration in the industry unless you have fundamental knowledge. So University of Hyderabad was so Whitehead had MIT, University of Hyderabad provided the backdrop, so it is a natural partner. That's what Dr. Prakash Babu was talking. It went through turbulence, because anything new will go through turbulence, particularly in a traditional country like ours. But it's like biology, survival of the fittest. This is Darwin's evolution. It's a selection process, right? So it has selected itself and changed avatar like our gods and goddesses, and has come to this stage today. And I've been at some level and party to all this. So kudos to them. A big hands to Srinivas, the young leader at the helm. And I hope this kind of, and interestingly, not too many of this kind of experiments have been done in India even after that. And correct me if I'm wrong. And it's in Hyderabad, which is a great center for which has both industry and academics combined, is one of the best place to work in India. So I wish it all the best. Thank you. Now, what I'm going to talk to you today is a story in my lab that is going on for last 20 years or so, and we recently had some nice success. So I will just give you a progress report and tell you in a story form, and please feel free to disrupt me and ask questions after my talk, or even if you are uncomfortable in the middle, and that's okay, right? Okay. So this has to do with GPCRs and membrane lipids, and I'll tell you why it's an important topic and how it evolved. It will also give you some idea, especially to new investigators. And this is a problem I see. I go to ISARs, and Prakash said that I mentor a lot of these institutes. Young people, science is incredibly complicated to do these days. There's impact factor, patent, funding. How do you find a niche for yourself if you're well trained and come from abroad in India to work with limited budget to do something that really be impactful science? Now I tried my way, and I'll show you my story and see where it is today. Okay, so this has to do with GPCRs, G protein-coupled receptors, which doesn't need any introduction in this audience. Enough to say that these are the major proteins that signal from the plasma membrane, And my interest grew in this because my background is in membrane biophysics and membrane biology. It traverses the membrane 7 times, not 1, not 2, but 7 times. Generally, the N terminal is here, the C terminal is here, and it acts as a receptor to various ligands, which could be a molecule. Any problem? Yeah. Which could be a molecule like serotonin, which we work with, but the ligand could also be a light. For example, we see this beautiful world, this beautiful campus through our eyes, and that is facilitated by a protein, by a G protein coupled receptor called rhodopsin. Now, rhodopsin is one of the early GPCRs that people worked with, including Hargobind Khorana, the Indian Nobel laureate. But the other GPCRs signal. Fine. Yeah. Thanks so much. Yeah. All right. Sorry for the interruption. So GPCRs work because the signal comes and binds to the receptor. Depending on which type of GPCR, it could bind outside the membrane, inside the membrane, and they activate a number of signaling pathways through G protein coupled G proteins. That's the name GPCR, G for G proteins. These are trimeric G proteins that you find only in higher eukaryotes, not in yeast or bacteria. Well, yeast has some G proteins, but not trimeric. And there's G alpha, G beta, G gamma. They get dissociated, and this signaling is very wide and varied. And so these, because of this very diverse signaling, they act as drug targets, right? So GPCRs today are, according to FDA, about 36% of today's approved drugs are GPCRs, which means if you go to a medicine shop and buy 3 medicines, one of them at least will target GPCRs, and this is not all. The future, and I see a lot of young people, is more interesting, because although GPCRs represent 40% of current drug targets, only a small fraction of all GPCRs are presently targeted by drugs. So about 830 or so GPCRs are there in your body. We carry 830 odd GPCRs, and only 15% of them are drug targets. So that means about 120 or so. What happens to other GPCRs? So it turns out there are diseases that are difficult to treat by today's so-called allopathic drugs, right? And there are these diseases that could be, now there's a lot of experiment going on in different labs around the world to see whether these drugs can be treated by some GPCRs. And there's another possibility, and this is something we are also working with. But lately we are working on GPCR endocytosis, although I won't be talking in this talk about that. Previously, people thought till 5 years back that all GPCRs signal from plasma membrane. But now we know GPCRs can signal from nucleus, from endosomes, from lysosomes. Okay, so that increases his drug targeting ability, drug target ability. So if I give this talk 5 years from now in the same auditorium, it is possible that about 60 or 70% of drugs will be targeted by GPCRs, and that's a very rosy scenario if you do GPCR biology, and it's good for good public in general, because more drugs means also more, and not only drugs, more refined drugs, because we can control GPCR signaling as biased and canonical to reduce side effect of drugs, and that's very important. And this brings to bench to bed. Okay, so we work with serotonin receptors. Serotonin, as you know, acts as a neurotransmitter in the brain, and is present in a variety of organisms, from nematodes to fruit fly to humans, shown in a cartoon here. But chemically, it is this kind of a molecule. It's like tryptophan, it's 5-hydroxytryptamine. Serotonin signaling involves cognitive and behavioral functions. So when signaling is not correct, you get in depression, anxiety, schizophrenia, OCD, and other problems. Now, when we started work in this, since CCMB had no receptor group, no neurobiology at that point, we had to wait a little to set up our lab while we were working on the other part of the lab, membrane biophysics. We looked at this molecule, and we have been doing a lot of tryptophan fluorescence to find out membrane proteins, how they interact with the membrane. And this I have been doing from my PhD and postdoc years in US. With that kind of curiosity, we looked up the fluorescence of serotonin. And to our astonishment and then happiness, we found out that neurobiologists did not know serotonin fluoresce. So at that point, we did detailed characterization of this fluorescence and published a paper in Biophysical Journal, and we even indicated how these fluorescence can be used for receptor work. So it's nice to be doing something that people haven't seen before. Now, like most neurotransmitters or receptors, serotonin has a bunch of receptors on different cell types, different tissue types, in which serotonin binds to facilitate different signaling, right? So same molecule serotonin can start or initiate different signaling because the receptors are different. So as of now, there are about 16 or 17 serotonin receptors, out of which 5-HT3 is not a GPCR family, it's ion channel family, 4 transmembrane domain, the rest are all 7 transmembrane domain. Our interest has been always on serotonin 1A, and I'll tell you why. Just one thing I'll tell you to especially young students, in spite of all this fantastic biology we have done all over the world, genomics and proteomics, none of the G protein coupled serotonin receptors has been, even today, purified from a natural source. What do I mean by natural source? Where it is in your body. So our receptor, for example, is mainly in hippocampus. It's also in frontal cortex, but mainly in hippocampus. People haven't been able to do because it's very difficult. The source is very low, and with the available biochemistry and biophysics that we have now, you can purify, but the few femtomoles you will get is no good for you. You cannot even do a decent silver staining gel or something. A lesson we learned the hard way when we tried to purify these receptors in late 90s, and that was with my student Shanti, who got her MSc from University of Hyderabad Biochemistry Department those days. Okay, why do we work with serotonin 1A receptor only among these families, besides the fact we can only do so much with a small lab? Well, this receptor is very interesting in the whole family of serotonin receptors. It differentiated at a time when vertebrates and invertebrates diverged. So ecologically or evolutionary, this has a very important point. On a more laboratory scale, how do you, if you cannot purify this receptor, how do you work with this receptor? This is your chemistry came in, and this molecule was synthesized by French medicinal chemists in early 90s. And this molecule you can buy this or synthesize this in this form or also as a tritiated form. So you could do radioligand binding assays, and this binds to 5-HT1A specifically with nanomolar affinity. So Kd is nanomolar. So you can easily separate out your receptor from a mess of cellular lysate or a crude plasma membrane just by doing ligand binding, and you can find out the functional state of the receptor. We have been doing that for years together, and we wouldn't be able to do this if this molecule was not there. The ligands to this receptor exert anxiolytic and antidepressant effects. So these are good therapeutic agents to treat neuropsychiatric disorders such as anxiety and depression. As the world is getting more complex, there are all kinds of psychological problems people face. Pandemic increased our load of anxiety and depression. So these are very good opportunity to work with these kind of receptors. This was among the first serotonin receptors which was cloned by Bob Lefkowitz, who actually helped discover GPCR and got Nobel Prize in 2012 in chemistry with his postdoc Brian Kobilka. And the human, rat, and mouse receptors are cloned, and the amino acid sequences have been reduced long time back. Mutant mice lacking this receptor was generated by 3 laboratories in 1997, and all the 3 laboratories are somewhere in New York. They published in the same journal, PNAS, and it turned out people expected that they will learn a lot of serotonin 1A receptor biology from this mutant mice, which they did by all the behavioral studies. But this is very interesting for drug companies, because when people, for example, work on cocaine addiction, seems to be this mice is a very good tool for novel drugs for cocaine addiction. So this mice has served as a very good tool for experimentalists in academia as well as industry for discovering new drugs in this area. So taking it all together is a good receptor to work with. This also works as an important drug target not only in neuropsychiatric disorders but also some types of cancer. So we stick to 5-HT1A. Our majority work is in serotonin 1A. Okay, so this is the hippocampus, hippocampi, 2 parts. There are 2 hippocampi in 2 parts of your brain. it stores your memory, it generates your memory, it stores. So if you have a injured hippocampus, you may lose your memory temporarily or permanently. For those of you who see old Hindi movies or Telugu movies, sometimes the hero loses memory because you're a motorbike accident. That's hippocampus injury. Of course, hero may recall memory by looking at a beautiful face. That we cannot tell. That is not from hippocampus. That's amygdala. That's emotion. Right, Professor? Okay, but hippocampus stores memory. Okay, so let me tell you how I got into this. I was happily doing membrane biophysics. I'm in CCMB, and every research council is asking me, what biology are you doing? You have very good biophysics, but what good are you doing for society? What good are you doing? And I was young, I didn't know what to do. But I looked at the literature and I saw this. If you look at GPCR, they are discovered in 1970s, and there are two names, Levkovich, who got Nobel Prize, Khorana, who already got Nobel Prize in nucleic acids synthesis in chemistry in 1968 or 69. I was in high school those days. Same year, Khorana got Nobel Prize, Neil Armstrong went to moon. I remember India was absolutely jubilant. We were just beginning to understand what he did at a school level. Khorana had a second career. Many of you don't know this. After nucleic acid, he realized membrane biology is a very toughest subject. He completely switched his lab in mid-70s with some Indian postdocs, Padul Chakravarti, C.M. Gupta, Radhakrishnan, and some American postdocs to look at lipid-protein interaction in membrane using initially bacterorhabsin, which is the bacterial version of a G protein, because bacteria doesn't have a G protein, and then rhodopsin. Both did pioneering work, and Khorana also did equal amount of work for getting a Nobel Prize. Unfortunately, he didn't get the second Nobel Prize, but Bob Levkovich did. But what was the work people are doing from 70s to 90s? If you, like I was looking in 95 or so, or late 90s, at GPCR literature, there are two kinds of work, mostly done in Europe and US. One was signaling. People like signaling. Biologists you can study MAP kinase, you can study cyclic AMP, you can study many kinds of, you can buy a kit and you can have a cell line and you express the protein in that cell line. The clone was available by that time, and we also did that, and you can do signaling. And that's very important. Many functions in many cellular background. Other was pharmacological, drug discovery. Lot of pharmacological people. This is more in US than in America, in India, but even in India, there is work going on in certain labs on some kind of a drug-related pharmacological profile. We argued at that point in time, this protein spans the membrane 7 times. So is it going to be very difficult to imagine the lipids modulate its function? Now, we knew this because we came from membrane biophysics point of view. We knew that smaller proteins like peptides or even channels like nicotinic acetylcholine receptor, which has 4 transmembrane domains, which I worked as a postdoc in UC Davis, does interact with lipids, and their function depends on lipids. And since lipids in your body are regulated by your age, and I remember Professor Subbarao in this context, who unfortunately passed away recently, who has done a lot of work on aging and that kind of stuff, and development and disease. And you can have a genetic mutation that you don't get the right lipid, and I'll talk about one such disease. Then what happens to those interactions? So our question then was, why it is a big question for GPCRs? Well, for soluble proteins, if you know sequence, temperature, pH, ionic strength, and presence and absence of denation, you can predict long time back, 20, 30 years back, you could predict its folding, at least in a test tube. Now, in a cell, it's complicated because it's crowded, but there are chaperones. And thanks to some excellent structural biology work, we knew by the early part of this century how those chaperones work. What about membrane proteins? They are about 1/3 of the proteome. Well, all these factors are also important because membrane proteins are also polymers of amino acids. However, these conditions are necessary but not significant. What will make it significant? Nature of lipids in close contact with the protein. So the same lipid and the same protein, if I have no mutation in the protein, but is there and there's a phosphatidylcholine next to it, and in another case there's cholesterol next to it, one may be active, one may not be active. You will not find that by sequencing the protein because there is no mutation, there is no covalent change. So can this happen for GPCRs? So membrane protein folding and therefore function depends on closely spread surrounding lipids. How do you look at them? How do you look at closely spread surrounding lipids which are not covalently bound, by conventional biological techniques. That's a problem. You have to do either spectroscopy or some kind of microscopy, and microscopy with very good resolution, which was not available those days. But you can ask a question. So, two questions we asked, because every project starts with a question. Which class of lipids interact with GPCRs for maintaining their function? Can we identify them? Can we characterize them? Second question, if we do this is basic biology. What happens to human health if the biosynthesis of such lipids is defective? Many of you know lipids, unlike proteins, do not have a genetic background. Lipids don't have a gene that codes them. Lipids are made by enzymes that have a gene, right? So lipids don't, that's why lipids don't have some kind of a genetic background. So let's say the enzyme that makes cholesterol in my body, some of the enzymes, there are 21 enzymes that makes cholesterol, is defective. And do I have a problem with interacting with my GPCRs? That was the question we asked at that time. And now I'll tell you where we are. Now, why cholesterol? Well, we took a shot at this after long. So cholesterol is an interesting molecule, a very small polar group, an interesting ring structure, which is only one double bond, but is almost planar. And chemists have worried a long time for this, why it should be planar. And there's a flexible tail. Now, it's a touch me not molecule, like some leaves you can touch and it will just retract. Any part of cholesterol, if you modify, is no more a cholesterol, and you can get into disease. So even if we add a double bond and take out a double bond in cholesterol, you get into fatal diseases that happens if the enzymes don't work very well in your body, like Smith-Lemli-Opitz syndrome. And we just had a paper in Traffic about 2 months back showing that in those patients, the GPCR serotonin is not trafficked right. It ends up in lysosomes, right? Only one double bond difference. So one double bond can make a difference between life and death. Okay. So on the top of it, our receptor was in cholesterol, was in brain, and brain cholesterol has some very unique characteristics. So although only 2% of your body mass is in central nervous system, 25% of free cholesterol is in brain. So brain is a highly enriched in cholesterol, like than any other part of your body, liver, kidney, any other part. Okay, so brain cholesterol is made in brain, body cholesterol is made in body. They don't crosstalk because there is something called blood-brain barrier. Although they occasionally crosstalk and people are still talking about it. Okay, people who work with stroke, for example, Prakash would know, have strategies based on that to treat patients. But we don't understand the science of that very well. Cholesterol does not cross blood-brain barrier, I told you. But defects in cholesterol metabolism associated with serious neurological and mental dysfunction, for which till to date there is no drug. Smith-Lemli-Opitz syndrome patients doesn't have drug. My student Yamuna Devi Paila and I used to work on this. We used to get samples from Radharani Devi from CDFD. And those patients, unfortunately, they will have some facial discomfort, they'll have cleft palate, but they basically don't have cognition. And the only thing doctors can do, feed them more cholesterol, so they have a more nice lifestyle, but they all die between 10 and 30, depending on his SLS1 and SLS2, all because in this disease, you don't eventually have more cholesterol. All of you know about disease due to more cholesterol, so-called cardiac problem, but less cholesterol. They have less cholesterol, more of the precursor, which is one more double bond. Okay, cortical cholesterol content has been shown to be lowered in mood disorders. This is from, of course, post-mortem studies. And statins, which has been mandated by FDA around 1990, is a blockbuster drug, one of the top 3 drugs in the world. Also produce things like depression in some patients. It is not our study, but this tells you why should reducing cholesterol make you depressed in some cases. And although this doesn't happen to a majority of population, it happens to a minority population. Those minority doesn't have the same genotype. They come from China, they come from India, they come from Palestine. It's not that only one kind of people. So cholesterol is a good thing to work in the context of neuronal membranes and neurobiology. Okay, so how do you handle those days we are doing hippocampal membranes. My student Thomas Pukadin, now a faculty in ISER Pune, who got Bhatnagar award a couple of years back, did the experiment. We take, and this is chemistry again, I go back to, you will see we keep on coming back to chemistry and biology. This is a carbohydrate polymer, a Heptamer, right? It's a cyclodextrin, right? It's a soluble protein. It's a soluble molecule, not a protein. So this sequesters cholesterol in 2:1 ratio if you allow it to incubate 2 membrane. The 2-to-1 ratio was found out by a very famous chemist called Breslow, who used to work with Columbia. My friend Uday Mitra worked with him. So Breslow showed this is 2-to-1 ratio in a Jack's paper in 1986, and we use them to suck up cholesterol from the membrane. If you do it in right condition, it will take up the amount of cholesterol that exactly you want. How do you put it back? You make these complexes, and then you incubate the membrane with this. Nice thing is membrane is hydrophobic. This is polar, so just a spin can separate the two. So you can nicely catalytically change, exchange cholesterol back and forth from membranes by doing this, and then you can measure how much cholesterol you have lost by doing a cholesterol oxidase assay. Cholesterol oxidase assay is the old assay. All we did is used an assay that uses a fluorescence marker as a probe that makes it more sensitive. You can do it in a test tube, or you can do it in a 96 well plate in a more high throughput kind of thing. And those things you can buy now from companies. It's more or less not so expensive. So that's what Thomas did, and this is how Thomas looked those days. Now he's, of course, very respected. He's a chairperson of ISAR Pune department in biology. Thomas showed that, of course, cholesterol goes down, but he showed for the first time that the activity of ligand binding and G-protein coupling also goes down. So you can see here the activity goes down to about 50% if you take out cholesterol up to 70–80%. This was now the first clear demonstration that membrane cholesterol is required for GPCR function, at least for serotonin 1A. We did not know at that point whether it's a general GPCR or only some GPCRs do it. We had no idea what the mechanism, but we had absolutely clean idea that this happens. Before our paper, you would see occasionally one or two people from Germany or one in US have showed one figure in a 9-figure paper showing lipids have some effect, but they thought this is an artifact. They didn't really report it very well. We went ahead and reported it and said, this is it, and let's now look into the mechanism. So mechanism, Thomas also showed G-protein coupling reduces this GTP gamma S, which is non-hydrogen analog of GTP. So ligand binding goes down, G-protein coupling decreases. Key question, this could be artifacts. You have to be critical of yourself. If we can put back cholesterol, do you get it back? Or otherwise for some other reason, not because of cholesterol. We put back cholesterol. It was not easy to do that in hippocampal membranes because it's very lipid rich, but we could do it. And we could put back 70%. We got back 70% activity back. In cells, CHO cells, fibroblasts, we could put back 100%, and we got 96% activity back. So yes, it's reversible. So at that point, we thought of mechanism. Now, whenever you think of cholesterol, you think of fluidity. So everybody told us that, well, you are changing membrane fluidity. Of course, you are by doing membrane fluidity changes. But we found out very soon, because we used to do membrane fluidity measurements ourselves. We have actually helped some, so had our colleagues. There's a colleague who used to work on urea toxicity, you remember, who passed away unfortunately. He used to come to our lab and do the work. And so we changed fluidity by not by cholesterol, but by alcohol or something. And did the ligand binding change, but not that much. So we said, well, this might change fluidity, but this is not the main reason. Physical is not the reason. We speculated that it binds to a certain regions of the protein, and that changes some secondary conformation, not global conformation, local conformation. But we have no purified protein. We cannot show what conformation changes. Who is going to believe us? Okay, how do you prove it? When you don't have purified protein and you are giving a fancy hypothesis, nobody is going to believe you, right? So we had to set experiments to show we are not wrong, although we cannot nail it down to the coffin. So what are we doing then? Well, how to test it? Series of students, initially Thomas, then Yamuna, who came from Vizag and Eluru, and then Sandeep, my lab member still now, designed experiments where if our major tenet, our hypothesis, working hypothesis, cholesterol interacts with the receptor. If we do not allow it to interact with the receptor, what happens to activity? Now, you can allow it not to interact with the receptor by depleting. Without depleting, you have to do. Well, you can oxidize cholesterol with something called cholesterol oxidase. Cholesterol oxidase is an enzyme, and it oxidizes the OH group to ketone group. It's no more cholesterol. But the physical property of fluidity remains the same. We did that, we measured ligand binding, it goes down. You can also treat with nystatin, which is a fungal antibiotic. Many of you have used the analog of nystatin when you have fungus infection. It's a bit like amphotericin B, but not exactly. Or detergents like digitonin, which is like saponin, they also, so both of these sequesters are complexes cholesterol. So you complex the cholesterol before doing the ligand binding assay, but don't deplete. So we did that, and the result showed our activity goes down. Finally, when we could heterologously express the protein in both CHO cells and HN25 cells, which are hippocampal neuroblastoma, we showed that when the cholesterol is less, just because you are inhibiting the biosynthesis, and you can inhibit the biosynthesis either the first step, which is called statin, is the first step of cholesterol biosynthesis. This is the rate determining enzyme, HMG CoA reductase. Statins inhibit this process, but you can also inhibit the last stage by an enzyme called by an inhibitor called AY944. Whether you deplete here, inhibit here or here, doesn't make a difference in activity. Activity goes down, showed by Yamuna and Sanjeev. So the take home from all these experiments, set up differently, is membrane cholesterol is necessary for receptor function, irrespective of the way you set up the experiment. You can set up by oxidizing, you can set up by complexing, you can set up by inhibiting the biosynthesis, doesn't matter. It needs cholesterol for function, period. Okay, now let's do the experiments in a more physiological way. Because MBCD is not in your body, it's a chemical, and it's a quick, dirty way to do the experiment, but that's not physiological. Also, body cholesterol changes much more slowly, it doesn't change so fast. So statins, I told you, was one of the best-selling drugs. It was, clinically approved in US at least in 1990. And it hugely changed mortality rate due to cardiac problems. Because 70s and 80s, there's a huge rise in junk food, mainly burgers and lot of red meat, and people are dying even in America a lot. So statins really stopped that to a large extent. One advantage of using statins is to lower cholesterol in cells, is that the process is physiological, it's chronic process. The cell is a long process. Even in cells, it takes 16 hours, 20 hours. It's not half an hour treatment of MBCD. So cell can do a resistance, right? Cell can give its own mechanism, while MBCD just physically takes it out. So these are the results, and you can see we only decreased 30% of cholesterol here. Still, we see both agonist and antagonist binding going by almost 50%. So what is the important thing here is for getting 50% agonist activity or antagonist activity down, we have to reduce 70–80% cholesterol by MBCD. Here, only 30% does the job. So take home from here, it is not how much cholesterol you have depleted that matters, how you have done matters. And that's the debate of acute versus chronic. In this paper, we actually hinted why people who take statin could get depressed. We didn't give a mechanism, we hinted in the discussion. Because statin is a worldwide, this paper was in ACS press pack, which means it went to all US newspapers and they interviewed us. We also got a lot of press coverage in India. Okay, now let me tell you where we are now. That was 2004. This is 2021. In our own lab, we have done 2 more GPCRs, and so the question is, do all GPCRs show sensitivity to membrane lipids, particularly cholesterol, or no? So there are 832 GPCRs in your body. So we have shown that adrenaline receptors, in collaboration with a group in JNU, and T2R4 receptor in a collaboration with a group in Canada, in University of Manitoba, they do. So this is our own work. But there are others who have been working, and there were 60 GPCRs, and this work has come from Germany, this work has come from Israel, from US. They have followed us, and they have shown that they exhibit cholesterol dependent signaling. Whether others do or not, we do not know, because people have done experiments. But there are receptors like secretin, cannabinoid, CB2, neurotensin 1, they do not exhibit cholesterol dependence in their function. So you can deplete cholesterol, nothing happens to the signaling. Now, what makes a GPCR sensitive to cholesterol and some GPCRs not sensitive to cholesterol is $1 million question now in this field. Okay, let me tell you a little bit about GPCR structural biology in the last 5 years, because that has changed the field hugely. Many of you know it's very difficult to purify membrane proteins, it's more difficult to crystallize and get structures because they're very floppy structures. If you try to take a picture of a person who is moving, you get a hazy picture. Since membrane proteins are dynamic, you always get not a very well resolved picture. That problem is being solved now in the last 5–10 years, and I'll tell you how it is done for GPCRs. So this is the chart. Class A, Class B, Class C, Class F GPCRs. first GPCR structure came in 2000 by a Polish man who lives in America. His name is Chris Palczewski in Carnegie in Cleveland. Now, after that, for about 7 years, there's almost no structures. One structure here. 2007, there was another big thing because Brian Kobilka and what was the problem? Problem was these are cryo-EM was not available those days, only crystallization. Very difficult to crystallize. You can do NMR, but doing solid state NMR of this protein is very difficult. But lately, you see the structures have gone up, and there's a huge amount of data now submitted. There are about hundreds of GPCR structures available now. What happened in 2007? Well, 2007 these structures came up. Sherezhov et al. Showed in a science paper that beta-2 adrenergic receptor, there's a receptor that that's affected when you take those puffs when you have asthma. They actually work on this receptor, beta-2 adrenergic receptor, and so they did a structure that is about less than 2 angstrom resolution. I was at that point visiting Belgium for some talks, and I got an email from my lab late night saying that it's some exciting structure, very good for us. What is good for us? There's cholesterol in the middle of the two monomers. Now, till then, people could not resolve lipids in protein structure because lipids are small, molecular weight 1000, 500. Proteins are big, so you cannot see lipids, but they are very good. 1.55 angstrom resolution, they could see lipids. Next year, the same group, well, one of the same group, showed same protein in different form can have cholesterol bound to helices. We have been telling ever since Thomas's paper got published that closely bound cholesterol dictates function of GPCRs, but we didn't have these pictures to show. These people show nice pictures and published in very good journals, but they are not actually in cellular membranes. There is something called lipidic cubic phase, which is not a physiological relevant membrane. But if you add their result with our result, you will see it makes perfect sense. So I visited their lab, they started collaborating with us a little bit. And then there are a whole bunch of structures come out, mainly from Ray Stevens and others, that shows many GPCRs that I showed you the structure is going up, shows bound cholesterol. So we did recently a statistical analysis published here. So 40% of unique GPCRs display bound cholesterol molecules, and 27% of GPCR PDB structures display bound, because one GPCR can have more than one PDB structure. So between 27 and 36% of GPCRs show bound cholesterol in their cryo-EM or crystallography structure. So it has become a hallmark to have some cholesterol in many GPCRs. Mind you, this is structure. People always haven't done the function. Function has to be done in a different assay, right? But it is important. Now, this is the story of the GPCR structural biology these days. You see the green is cryo-EM. Cryo-EM is slowly pushing down crystallography. Because cryo-EM is very good for GPCR structures because it is very good for complexes. Cryo-EM is very good for big structures. And GPCRs often dimerize, oligomerize. So it's a big thing. Crystallography is good for small molecules. Cryo-EM is very good for ribosome, for example, that Venki Ramakrishnan did. So cryo-EM is slowly pushing. This year, you see cryo-EM is hugely more than crystallography. It's ongoing data. Okay. Now, if you see how many cholesterols are bound per GPCRs in the molecule that has GPCRs, you see it varies. It could be 1 to 10, and there's 116, but this is actually misnomer because this is a dimer. So actually, per monomer is 8. This 10 actually is from our receptor. The structural serotonin receptor came this year in a Nature paper in March from a Chinese group, and we are very eager to see how many does it bind cholesterol, and if it does, how many? It's 10, and I'll show you the structure in a while. So at that point, when all this was coming out, we started thinking, if cholesterol interacts with GPCRs, does it interact with any place, or there are some cholesterol binds? So he looked up sequences, and he found this sequence called crack, which people showed before the soluble proteins at least, like caveolin 1, binds cholesterol through the crack domain, and crack is just a tyrosine, in arginine or lysine, leucine or valine, and some amino acid in the middle. So it could be as short as 5, as long as 30. So do GPCRs have crack? So when we did this, people have worked in all these proteins, channels, but there's no GPCR work at that point. So in 2011, I think, so how do crack interact? This is the theory. Valine leucine binds the isoactive chain because they have Van der Waals interaction, nonpolar. The tyrosine interacts with the ring, which is a double bond, and lysine and arginine interacts. But this is a hypothesis. There is no proof of this. So first thing we showed in 2011 is that yes, major GPCRs that show cholesterol dependence has cracked sequences, which doesn't mean they are binding cholesterol. At least in the sequence you see cracks. Our serotonin 1 receptor has 3 cracks, all 12 units. One is transmembrane helix 2, one is 5, one is 7. Now we have to prove, are these responsible for the cholesterol sensitivity? And we did 2 approaches for this. One is simulation and one is mutation. You mutate these sequences and see which mutation shows cholesterol sensitivity, which doesn't. So I'll show you results from both. Simulation, we do coarse grain simulation using Martini model, and our collaborator is Dr. Durba Sengupta from National Chemical Laboratory, Pune. Actually, we started collaborating with her when she was a postdoc at Denmark, at Netherlands, and then she got a job in NCL. It's very good because when we started 10 years back, 2011, 2010, to simulate a huge molecule called GPCR, which is typically 400, 500 kilodalton, often in oligomers with water, sodium ion, is very difficult for our kind of computers to do. So this coarse grain does atomistic, but 4 atoms it pieced together and you get a long-term simulation. So we have done a lot of work, and I'm just summarizing it in this slide. We have shown that cholesterol binds to some hotspots of these proteins, and this is serotonin 1A, but it binds, it also unbinds, it goes back. Some exchange is fast, some is slow, but they are basically microsecond. So our major conclusion with work of last 10 years is microsecond time scale is dynamic, is weak binding, but it's crucial. So at any point you have to find out how many cholesterol are bound, how many are going out, and we actually scaled it up and showed how to measure it. So the energy minima is kind of low. In the cholesterol occupancy, the energy landscape corresponds to a series of shallow minima interacted by low energy barriers. So it is not that they are bound very tightly. So the word binding, if you say in biochemistry, people think it's not very tight binding. It's quite labile binding. So Durba and we decided we'll show it like this. If a cholesterol molecule is present during our simulation is 3 microseconds, at one spot we'll call it 100% occupancy. So these greens are the crack sites. You see in one crack site, transmembrane helix 5, it is present. But there is this one, you see, there is no crack site, but you see 100% cholesterol occupancy. So you cannot decide by just looking at crack sequences that they are responsible for cholesterol sensitivity. And neurotensin 1, which I showed you doesn't show cholesterol sensitivity, these authors are Tony Watts from Oxford Biochemistry. They showed in the paper, they wrote in the title, it has crack domain, but it doesn't show cholesterol sensitivity, its function. So what decides then? So what we did then is this. We thought we'll mutate. So we mutate, we produce both single and double mutants. So tyrosine, we mutated to this lysine to alanine, and so on and so forth. We also did double mutations. We took transmembrane helix 2 because in transmembrane helix, this helix and this helix, there's some functions that are compromised. We didn't want to do that. There's an NYX motif there. Okay, and so we did this and we expressed them in cells. It was a lot of work. We are helped by Dr. Panikkar of NCBS and his students. And this is what we found. Now, what is the signaling you're looking at? Remember, serotonin 1A in normal membrane, hippocampal membrane, is a GI protein, GI coupled protein, inhibitory G protein, right? Which means cyclic MP goes down. Stimulation by agonist reduces cyclic MP. We measured that. If you stimulate the receptor with his agonist, cyclic MP goes down. Since going down is difficult to measure, you put forskolin, it goes up. Then you see how it goes down. So we do the same assay, and these are the students, Aditya and Parijat, who did that. Aditya just went for a postdoc in Michigan. Parijat is still there. He will go in somewhere next year. He's also done. So this is our wild type. Wild type, you see, if you deplete cholesterol, signaling goes down. Reduction in cyclic MP is not there. That means it's sensitive to cholesterol. We know that. Among all these mutations, everything showed like wild type except 2. And in both these 2, there is K101. If you touch K101, lysine 101 transfer helix 2, either single mutation or double mutation, whether you deplete cholesterol or not, you see here and here, this is no significant difference within these. So this receptor doesn't sense cholesterol if you mutate K101 to alanine. So this is the key to cholesterol sensitivity, at least in serotonin 1 receptor. So this is our conclusion here, lysine 101 in transparent 2 confers cholesterol sensitivity. Okay, now this was published recently in Science Advances, and we also did simulation with Jana Salent from Barcelona. And so this is the recent structure that has come out in March from this group in China, published in Nature, and they showed there could be, they reported 3 structures. In one of the structure, there are 10 cholesterol molecules bound to serotonin, 1 molecule of serotonin 1 receptor. Now, this is the APO receptor, which doesn't have the ligand bound, and this is the maximum number of bound cholesterol molecules reported in any GPCR structure till date. We are quite happy because this was the first receptor GPCR that maximum amount of cholesterol sensitivity work is done mainly from our lab. So it matches structure and function, at least matches in this case. Now what these people do, we did simulation in this case at atomic scale, and they show cholesterol actually binds tightly between transfer helix 1 and 2 by establishing polar contact with K101, which is shown here. So remember I showed you a cartoon initially that the lysine interacts with cholesterol by polar group. They actually showed that. As it so happened, about 1 month or 2 months after this result we had, but we haven't published the paper, this paper came out, cryo-EM, and simulations show this position of cholesterol, show a tightly bound cholesterol in the position almost identical to the one observed in the recent cryo-EM structure. So there are 10 cholesterol molecules bound in the serotonin 1 receptor in the cryo-EM structure. One of them is exactly here where we are predicting, and that's the one we think is giving the cholesterol dependent signaling. So this paper came out about 3 months back and is recommended in faculty opinions. But this is only for our serotonin 1 receptor. What about other receptors? Well, we are doing some work with collaboration with other groups, but I think it's really, you really have to see. So if, let's say, natural mutant doesn't have this lysine 101, then a lot of signaling will not be cholesterol dependent, and that's the implication. I have to stop because my time is almost up. This is my group, more or less the same group, 1 or 2 people have left. This is my ex people who have worked. This is Thomas, this is Yamuna, these are some others, Shanti, and we have very nice collaborators. I didn't talk about all of their work, but we get help from many people from India and abroad. Sadhu is in Cleveland Clinic, Andrew is in Australia, Durba is in Pune. Dr. Panika retired, but he helped us a lot when he was in NCBS. We do single particle tracking with her, and this is Jana Salent. We get money from various sources. As you know, money has no color. This work is done in CCMB. We all know that. Little bit of advertising. We just got some new grants. So anybody who is interested can write to me, and I'll be happy to have a discussion. Thank you very much. I'll be happy to answer questions. Thank you. Questions? Yeah. The receptor itself. Yeah, membrane fluidity, when you say, you generally talk about mobility of lipids, segmental dynamics of lipids, and you measure by fluorescence, by DPH, or by NMR. You don't talk about the proteins generally. Crowding by protein can change membrane environment, and there are a lot of experiments people are doing that now. People don't call it fluidity though, the density and other things change. But fluidity generally is influenced more by lipids. In fact, a lot of people in neurobiology, neurochemistry, they do membrane fluidity experiments by just measuring the dynamics of a small molecule in. So probably not.
Because earlier in our system, in our lab, so when Shravan was under Dr. Sanjeev Jain, so
Please use the mic, yeah.
He carried out one experiment in pancreatic beta cells, so where we saw the cyclic AMP generation. So it had the presence of cholesterol in the membrane has reduced the cyclic AMP generation in those cells. So in that way, because you have shown in beta adrenergic receptor, there is decrease in cyclic AMP. So I wanted to understand that if you
Sorry. Beta adrenergic receptor is not a GI receptor, it's a GS receptor. So adenine cyclase CMP goes up. Serotonin is GI, right?
Yeah. So in that case, if you overexpress that receptor in a system, so does it change that paradigm?
Very good question. If you overexpress a receptor in a non-natural system, people always do for purification of proteins and all that, you have induced crowding. Now let me tell you this, we don't overexpress, we just express. We actually measure how much we have expressed by doing ligand binding and quantitate. We generally have 10 to the power 4, generally 10 to the power 5 cells per, 10 to the power 5 receptors per cell. People overexpress to do structural biological work because they need the protein. But overexpression has other problems. It can give you background signal, it can give you a signal in freight, for example. So all our experiments are done with non-overexpressed proteins. Yes, overexpression induces artifacts, you are right. So most overexpression is used for structural studies when they actually need the biomass. But for functional studies, if you overexpress, you can actually induce signaling that is not there to start with. So we don't do that. We actually, if we read our papers carefully, we do write in our materials method how do we measure receptors, and they are within a level. In case they are not, we fact sort them and make sure they are in the same range. Okay. Yeah, question there. Rahul. Yeah. Please put on the mic.
What is your take on that? The GPCRs which are cholesterol dependent and some are not dependent. So is it the membrane lipids also would be the major part? Only lipid would also be the important key factor which can have a function on that.
Yeah, it's a
Any clue they interact?
right. I get what you're trying to get at. So it's a very interesting question. Now, it may appear from my seminar that the only lipids GPC interact with cholesterol. That's not correct. In our own lab, we have shown sphingolipids, GM1, for example, is very important for this interaction. Brian Kobilka has shown in a latest paper, phosphatidylene inositol interacts with beta-adrenergic. So cholesterol, because we started our work in cholesterol, it has become very popular. But there are other phospholipids. For example, this March structure of serotonin shows bound PIP2 molecule, PIP2, phosphatidylene inositol diphosphate. That's a signaling lipid actually, right? So the whole repertoire of lipids, lipidomes, that interact with GPCRs are just slowly exploding. We know more about cholesterol because of our work and some other groups' work. But you're absolutely correct. It is also possible that there could be compensation or synergy between cholesterol and other lipids interaction. When you increase one, the other decreases and things like that. But we are not in that stage. No data exists that. But there are some groups, including ours, we are trying to do simulation with that. But explain later, no, not available yet. But good question. Thank you.
One more thing, sir.
Yeah.
So when the molecule will bind to the receptor, there will be conformational change and then the beta gamma will be dissociated. So is it the conformational change also is the important in the cholesterol binding, or is it the major factor in this?
Very good question. So if you see the March paper of Zhu et al. Nature, they have three structures. There are three structures. All the three structures have different number of cholesterol bound. The APO structure, which doesn't have the ligand, has maximum number of cholesterol bound. So yes, the secondary structure changes that are induced by binding of a ligand does make a change in cholesterol binding property. You're absolutely correct. Yeah. Yeah, Professor Anna. Mike, please.
So why the depression and anxiety disorders are less or not there in all statin users?
This is difficult to say, but I can tell you why we wrote in that paper that serotonin is a happy receptor, serotonin is a happy molecule, and we showed in that paper that if you deplete cholesterol using statin, at least in a cellular model, we showed that signaling goes down, serotonin signaling goes down. Now, why should that happen in certain population in a particular genotype and not happen to others? It's very difficult to say, but there are clues. One clue is, as I said in the beginning, and I refer to Professor Subbarao, lipid profile in your body changes with development and aging. Most tissues in your body, most, including brain, has more cholesterol as you age. One good example is alpha-crystalline. My good friend and colleague Mohan Rao worked a lot on alpha-crystalline, which is a soluble protein. We all think it's a soluble protein. It's a chaperone-like protein, right? But if you take an older lens, you will see alpha-crystalline is bound to membrane. It's not an integral membrane protein, but it's bound to membrane. A European group showed that's because older lens have much more cholesterol, and alpha-crystalline binds the cholesterol. So the topology, the distribution, and function of a protein can change in aged versus neonatal. Also, now people can do very good lipidomics, and people have shown the complete lipid profile changes with age. Okay. Remember, Alzheimer and Parkinson disease happens generally to older people. You will never see an Alzheimer patient, Parkinson patient who is 30-year-old or 25-year-old. And that's because there's a huge cholesterol connection in this. So all these proteins are not basically membrane proteins, but they go to membrane targeted by more cholesterol, A-beta 42 for example. And if you see literature of Alzheimer's, Parkinson's now, 50% of them has to do with cholesterol. So this developmental regulation of cholesterol with age, particularly in brain tissues, has a huge relationship with pathogenicity and other things. Okay. Second thing is, when people take statin, so statin we take orally, it should not go to your brain, right? But there are studies shown, clinical studies in England shows that statin takers suffer from less plaque kind of disease, less all the way. If you take something orally, cardiovascular system, why should it go to brain? Now, people are starting to believe that blood-brain barrier is generally tight, but in some cases it gets leaky. For example, when you drink alcohol, okay, and it's leaky for a small time. And there are some surgeries in neuro stroke-related context where people, surgeons temporarily compromise the blood-brain barrier, and they do the surgery. So naturally, also blood-brain barrier apparently gets, it's a very hot topic, leaky. I tried with Kiranam, didn't work very well, to look at blood-brain barrier in zebrafish, because zebrafish is a good model, and we tried to see by doing mass spectra something in zebrafish how much of cholesterol goes to blood-brain brain. Somehow for very different reasons we couldn't do the whole project, but it will be very good because there is no good model for blood-brain barrier regarding cholesterol as far as I know in rodent or something. It's very difficult to do. Zebrafish is easy. You can see under microscope, and there's a process called decoordination where you can increase that by, correct me if I'm wrong, 3 days or 7 days, right? So you can actually make zebrafish transparent for about a week just by using an agent. So you can do beautiful microscopy, and hopefully we'll do sometimes. But thanks.
Actually, mannitol can be given to damage blood-brain barrier.
Right. That's correct.
Blood-brain barrier damage also can be studied using Evans blue.
Right. That's one compound. The other compounds also, yeah. And there are some surgeries now that actually intentionally compromise blood-brain barrier for a while. Okay. But it is done by highly specialized neurosurgeons. Blood-brain barrier exists because that's basically astrocyte membrane. So cholesterol cannot go through a membrane because it's hydrophobic. But there are some ways to do which are very transient, and we still don't understand. It's either biology or chemistry. Any other question? Yeah.
I have a question. I think this is with reference to Professor Preslow's work.
Right.
Even Professor Jain carried forward a lot of this.
He did a lot of work on complex, clathorate, yeah.
On other folic acid based work.
Right. Uday Moitra also did that, yeah.
My
Methyl beta cyclohexane, yeah.
It has a propensity to pull out cholesterol and also be able to do that. Now, one thing in pharmaceutical, it is very broadly accepted that alpha cyclohexane, beta, and to some extent some of the analogs are well trusted, GRAS-listed excipients for a variety of drugs. Now, I was just wondering whether there would be an implication on eventual drug profile or any other side effects if they are able to essentially leach out cholesterol, because we are not looking into that from that angle.
They are not looking from that angle.
Second thing is, I guess,
And in medicine preparation, they use it. You're right. Yeah.
and even if you take a classical case of remdesivir, which became popular in the COVID, the typical formulation was something like for every there's a 20-fold or a 30-fold, 25-fold by weight of the excipient, which is actually cyclohexane-based derivative. So I was wondering whether we are overlooking something along those lines.
I have the same suspicion. You're right. So we actually realized this when we are patenting some of this. Yeah, MBCD comes in just like some kind of cyclohexane. I don't see any literature, you know more than me in this area, where Japan has done a lot of work on cyclohexane, Japanese pharmacologists, but they don't look at these aspects. They are basically pharmacy kind of people.
More of a, it's a solution aid.
It's a solution, yeah. So it's worth looking at. I haven't seen any literature based on this.
The other thing is,
But the formulation has it, yes.
formulation has it.
Yeah,
It's far more than the drug concentration.
yeah.
20, 10-fold.
Yeah, you're right. Thank you. Yes.
Sir, good lecture, sir. Are there any records about relation between hyperlipidemic patients and serotonin 1A activity? Are there any records so far?
Very good question. The answer is there are medical literature showing that yes, there are records. I'll tell you what kind of records. There's a record from Chicago that people, and this is post-mortem brain analysis, people who died out of depression, so people have taken serotonin 1A from those post-mortem brains, their ligand binding is not efficient. So the A is gone. Now these studies can only be done in post-mortem brains under certain conditions. You cannot do it here, you need permission. So there is a lot of studies showing agonist efficacy versus depression of in a post-mortem. In living sample, they are difficult to do because of ethical reason. Animal models, there are some people who have done that. Actually, more data is on human than animal because of this disease perspective.
Yeah.
There's some data. There's a person who, Prakash Babu may know, there's a person who is working at Ghanshyam Pandey, who is at University of Illinois, Chicago. He showed that 5-HT2C, which happens in platelets in blood, can be a good indicator of a person's indication to commit suicide. He actually patented this also. So there are a lot of indicators of serotonin receptors and mental state. Okay, and if you put cholesterol in the thing, but unfortunately, data is scarce because for a long time, this is generally done in medical schools and they don't talk much to people like us. I generally go and give talks there, so I know this, but I think we need to look more into this. You know, Prozac is a drug that many American athletes take to make them feel happy. Some of the presidents also used to take Prozac. Prozac is nothing but it acts to, basically if serotonin is less in the cleft, synaptic cleft, you get depressed. So all these antidepressants that you see, fluoxetine and all this, they are basically inhibit serotonin transporters, okay, and so they don't allow serotonin to go away from the cleft. So you need to maintain a minimum concentration, optimum concentration I should say, of serotonin in the synaptic cleft, which is typically 50 nanometer. So such a emotional and personal thing like depression, it relates to concentration of serotonin in the cleft. And most drugs, 50% of drugs or antidepressants is based on this. And Prozac is a commercial name. It's freely available in America. Lot of sportsmen has been disqualified because they have taken Prozac and then taken it. So yes, there's a huge connection with psychology. And there's a person in Nimhans, his name is what's his name, Sanjeev Jain. He actually works on, he's a psychiatrist, but he works on depression and serotonin gene. He works on the genetic level, things like splicing. He gets those genes and this is done. He's basically a doctor, but he has collaborations with people in IGIB and other places. So yes, but more needs to be done, that's for sure. And as Professor Randa said, if there's a particular genotype because of our diet or vegetation, we don't know that. There's not enough samples from India, unfortunately. But that's true in many other cases also. It's true for cancer also. Thank you.
Sir, and do you think that cholesterol has an effect on the structure of the protein because its ablation is...
So that's what I'm saying.
Yeah.
It has an effect on local structure of the protein, not global structure.
Okay.
Right.
So do you think there is a possibility that the cholesterol binds with many other membrane proteins as well?
Yes. But that has been shown. For example, nicotinic acid receptor, which we worked with, we have published this. I'm not an author, but one of my colleagues is, that if you reconstitute the channel protein with liposomes that has cholesterol and that doesn't have cholesterol, and since it's a purified protein, you can do circular dichromosome or FTIR, there is different percentage of alpha helix, 6, 7, 8% difference in the two cases. So yes, a lipid can change folding of a protein even if there is no mutation. This reference I have. If you write to me an email, I'll be able to give you that. Okay.
Thank you, sir. Very nice lectures.
Thank you.
Thank you, Professor. So it's a wonderful lecture and it's a lot of learning for each one of us, though we are not from GPCR fields. I have a question on the last part of your topic, where you mentioned about those crack components in the receptor, right? And I think for the receptor you are working with, there are about 10 cholesterol molecules bound. And
in one of the structures.
In one of the structures, that's solved by Zhu et al., I think, in Nature paper.
Right.
So is it known like one crack motif binds to how many cholesterol molecules? Because I think there's a clear disparity among the crack motifs. Of course, you also hinted that there may be other crack independent motifs as well, and that's one thing. And related to that, you mentioned about one receptor which has crack motif but does signal through that.
Right.
Do you see that under any specific circumstances, maybe a binding of agonist or something else, actually make it more cholesterol dependent?
Very good question. Thank you, Kishore. So thanks for asking the question, because I can mention this in detail. So although crack is a very well-known motif, there are other motifs that are suggested for cholesterol binding in GPCRs, other proteins. One is CCM that was suggested by Brian Kobilka and in the 2007 structure in the science paper. They have sometimes overlap with crack, sometimes they don't overlap with crack. But we wrote a review last year, and I can send you that. There are 3 or 4 such motifs present in membrane proteins. Whether they act in synergy or they act individually, nobody knows. And also, as you said, some of the proteins have this sequence, but they don't bind cholesterol, like neurotensin 1 has the sequence, doesn't bind cholesterol. So that's another question. But yes, there is a divergence of those sites, and in fact, in our title we wrote about that. We said that is it a slippery slope, because many people think you have a crack motif, you go mutate, you will get function. That's not true. Actually, we had to do 10. Initial 7 gave us nothing, and we are not a lab which does mutation every day. We had to work hard to make those mutations, and eventually we got lucky. We got 2. So it is very difficult to predict structure function by just looking at crack sites and this thing. But some of these sites like CCM has overlap with crack also. So many of us think that it's a combination of the 2. I can send you the review. It's very nicely written.
Cholesterol binding motif, which is required for the activity of some G protein receptors. Sir, is it possible that the motif that you are talking about is not exposed in the molecule which have that motif but do not show what is that? Means if cholesterol is present or not. Like
The-
for example, in tertiary structure, the cholesterol motif is exposed. That is why cholesterol is binding there. But in other molecule, it will not be so. But because of some, resemblance with the other molecules.
Yeah, it's a very good question. Thanks. That's a very nice possibility. The word you are looking for is latency or cryptic nature. Membrane proteins have topologies, right? So folds. So let's say the crack motif is inside the membrane, it will be accessible to cholesterol. If it is outside, cholesterol may not go there. That's the thinking, right? That has been thought, and some of that, surprisingly, if you do analysis, and we have done some of these analysis, if you do bioinformatics analysis of this crack in different GPCRs, not only serotonin 1A, you will see some of the cracks are outside the membrane. When you see a crystal structure, and when you draw a 7 transmembrane domain, where the membrane ends and the cytoplasm begins, is actually an imaginary line. It is not given by crystal structure. Crystal structure gives you relative orientations and lengths. That is done by hydropathy profile and all kinds of things. Second thing, from simulation we know, like our receptor has a big loop called intercellular loop 3, ICL3, a very floppy loop, which actually crystallographers cut before doing crystallography and put a antibody there. That loop every now and then goes to the membrane. Now you cannot see this in experiment, but you can see this in simulation. Now, what is the frequency of going, and is it enough to make a biochemical change? We do not know. But every now and then that floppy thing goes up. It's like a big loop, and this is the protein in the membrane. Suddenly it goes and binds there, stays 5 microseconds, comes back. Now if that happens, and if there's a cholesterol, it will bind. It can stabilize it. That's the exciting possibility. We are looking into this. But experimentally, how can you do this? Because all our biochemical experiments are time averaged experiments. It's very difficult to do biochemical experiments in microsecond time scale. So yes, there is a possibility of that, but it is, more work needs to be done on that. Good
Sir,
question.
Just curiosity.
Yeah.
Like we already know all the topological profile of cholesterol molecule. Means like where it is hydrophobic, hydrophilic, and all those things.
Right.
Sir, would it be possible to replace it with some other molecule which has the structural property of cholesterol, and then let it bind to the means protein, and see could it means increase the basal activity of enzyme?
Very good question again. So this is what I didn't elaborate on this because the lecture will be long. I told cholesterol is a touch me not molecule, and in my slide you show fine-tuned molecule. What I mean by that is if you have one more double bond or one more methyl group, it doesn't look like cholesterol at all. Okay, now there is a man called Conrad Bloch who discovered that cholesterol biosynthesis pathway in Harvard University. He was an organic chemist, but he knew biology very well. So he has proposed something called Bloch hypothesis. Bloch hypothesis says cholesterol is a molecule that synthesized. Organic chemists find it a fascinating synthesis. Biologists find it a very nice evolved molecule. Remember, bacteria doesn't have cholesterol. Yeast doesn't have, they may have cholesterol, like mycobacterium has cholesterol, but it is not made, they cannot biosynthesize cholesterol. They get it from environment. Insect cells like mosquito, Drosophila don't have cholesterol, but they get it from the auxotrophs, they get it from diet. Okay. So you need to be higher eukaryote to make cholesterol, which is made by enzymes in endoplasmic reticulum. So Bloch hypothesis says on one hand cholesterol synthesis is getting complicated, organic chemistry wise, on hand evolution millions of years. So cholesterol is a fine-tuned molecule both by organic synthesis, where is a double bond, where is a chiral carbon, to evolution of these two, the marriage of these two is cholesterol. So if you change cholesterol a little bit, it is no more cholesterol, and the difference in terms of biology could be life and death. So the answer is no, you cannot do that. We as fluorescence spectroscopists use fluorescent analogs of cholesterol. But each fluorescent analog has a probe that is little big probe sometimes. We need to do the test of that. We need to show by control that it doesn't affect the activity of the enzyme or the protein, or sometimes it will not actually affect the activity, but it will affect the trafficking. So you can also do trafficking. Cholesterol has a path by which it goes inside cells and comes out. Does it follow that? Some probes follow, some probes don't follow. And there's a huge literature on these things. People use probes because we want to do confocal microscopy. But cholesterol is not a big molecule. You cannot use antibody or GFP tag. So the answer is no. Most of the analogs of cholesterol look like cholesterol, or people think it looks like cholesterol. Cannot support all the biological activity of cholesterol, and that is fact.
Either we can think of deteriorated, but it will not show.
Deterioration is a little better, yeah.
Clinically easier, but it will not show in the dynamic time space what you are mentioning.
Right, right.
Because there is no.
And perturbation is less.
Yes. Appreciation could be another issue, but it makes it.
Appreciation, yeah, yeah. That will change the rate of reactions though because of the mass, but it will not change. Yeah, you are right. So people have done Dietary Nmr with dietary cholesterol. It's a little expensive, but people have done. But their sensitivity comes. NMR, you need milligram of protein. The milligram of membrane protein, nobody is going to give you, right? So that's a problem. But fluorescence you can manage with microgram, nanogram.
In case of psychiatric disorders or in depression or anxiety, did someone report the changes in cholesterol binding to the serotonin receptor 1A? If there is a change, that might change the fluidity of the membrane. That's why they have intensive signaling of some sort, thereby showing a psychotic disorder or anxiety or depression.
Not the way you are saying. No, it's not one-to-one like that. It's difficult to do also that kind of experiment because of human sample. People have, there's old experience. People have fed rat with different kinds of, food and looked at the micrometric membrane and things like that. Those are old studies. But once you cut a rat, it's difficult to. There's a lot of perturbation going on. It's difficult to measure fluidity in an intact rat. You can image the rat nowadays. There's not a very difficult thing to do. But that image's resolution is not as good as doing microscope in a polarization or something like that. So that's a problem. There is a new method called generalized polarization. At least it has been done for zebrafish at least, and hopefully also you remember any microscopy or spectroscopy you do, the sample has to be transparent. A rat is not transparent, right? Zebrafish is. So that's a problem.
Excellent. So we haven't heard that many questions in a long time. That's so nice to hear.
Yeah, thank you.
Thank you, Professor Chattopadhyay.
Yeah, thank you for all the questions.
Thank you for coming here. I mean, I know this was rather us, and thank you to the guests also who came at short notice. And you'll be happy to know, so we have been streaming your talk, and apart from the technical glitch, it went okay, and there were, I think, a maximum of 5 viewers from outside who were there throughout. In the beginning, there were 10, 15, and then it kind of changed. And Professor Abhijit Mitra from IIT was there, IIIT was there throughout, but nobody asked any questions. We were monitoring that, but you had enough questions here, so that's great. And so, yeah, listening to your talk was wonderful. I'm sure all of us enjoyed it because, the questions were great. It was truly interactive, and it's a great example of, I think, what we are trying to do with this seminar series. This will continue, so, it was a wonderful start.
Thank you.
And it reminded me also of my days in CCMB when we used to hear you talk and all of that. So thank you very much, and I request our director to hand over just a token of our appreciation.
So we wanted to go with an eco-friendly.
Yeah, that's nice. Thank you. Good to have age of this. Nice.
So we wanted to go on echo because it's been.
It's very good. It's very good. Thank you. Thank you. Go to the box there.
On those we'll discuss.
Yeah. Yeah. Can I get the USB?